Okay, so I'm going to run through how to produce a sterile inoculant with the minimum equipment that you can use to get a successful product. First step, you would start with a clean culture of the right species of bacteria for the target plant that you're aiming for. And to use that effectively to produce a large volume of inoculant, you would use a dilution technique. So you take a small volume of the bacterial culture and dilute it with a much larger volume of either a weak nutrient solution or something as cheap as deionized water. So to produce those two things, you would grow the bacterial culture up in a small pot. Here we're using a Duran bottle, which is small enough to be sterilized inside a domestic pressure cooker. The flask is then inoculated with the bacteria so that you would have this pot full of a cloudy bacterial culture. This is then connected to using a sterile connection. This is just an example, but you would connect that via a sterile connection to a filling spout. The other part of the setup here is a separate pipe which would go to a much larger reservoir of liquid. Here we're using something that's just a 10-litre carboy. So that would simply just suck the water up from, or the nutrient solution up, and give you a dilution from a much smaller volume of bacterial culture diluted down with the nutrient solution. So you can see they will both flow through into the same pipe. Here you have an aseptic transfer, and here you have a pumping system, which we haven't got in this mock-up, but you would be pumping the liquid from the larger container and pumping into that stream the bacterial culture. This flows through to a filling point, and here we're using something as simple as a stainless steel pipe which has been fashioned into a point so that you can pierce the plastic bag and allow the culture to pass through into the bag. So the second part of the process is to have the carrier material produced, and you would have in this case a dry powdered peat which has been milled into a fine powder, put inside a plastic bag. And then this whole unit has been gamma irradiated, so you have a sterile unit. Using the pumping system and the bacterial culture, you can then fill many hundreds of these packets using a very small flask of bacteria. And that's done simply by piercing the plastic bag with a needle, and the pumping system would force into the bag the diluted bacterial culture. Once the culture is inside the packet, the hole that's left by the filling nozzle is sealed just using a plastic sticker, and that gives us, in theory, a sterile produced peat-based inoculant. The bacteria then grows inside the packet, giving a final effective concentration or number of bacteria. And the way of producing is quite common here in that you use a blank bag which can be filled with any type of bacteria for different legume crops. Once you have your soya bacteria or your lupin bacteria, your clover bacteria inside the packet, it then goes into another outer packet which has specific instructions for how to apply it for the different crops, can have different brand names for different customers. It makes the basic unit that the product's made in much more flexible and saves on costs when you're producing for different end users. The farmer then applies it as though he was sprinkling the peat onto the seed. The way we produce it here is that we have a sticking agent inside the packet with the carrier material, so that the peat actually sticks to the surface of the seed as the farmer mixes it through. And this allows the seed to take with it the bacteria into the furrow, into the ground, and as the seed germinates, the bacteria are ready to infect the roots. So if we move on to a slightly larger scale of producing, we have here a 1,000-litre container of deionized water and a piece of filling equipment which is designed to fill sachets aseptically. So again, the same principles apply. We have a much larger volume of dilute nutrient solution, a small volume, or relatively small volume, of bacterial culture, and this passes through a peristaltic pump which maintains the sterility of the material in the filling tube, and this passes into the pouch filling station, which doesn't rely on the operator producing a sterile transfer by piercing the membrane. it actually uses a filling gland which is part of the pouch, and this removes any operator error. We can produce liquid products, we can produce surprisingly a granular product, and we can also produce the basic peat sachets that we've shown earlier.